Good morning. It's 9 a.m. here in San Diego, so we will begin the webinar. Thank you very much for joining us. So today's webinar is on linking protein structure to sequence and alignments. My name is Andrew Ori, and if you have any questions after the webinar, please feel free to email or call. During the webinar, you can ask questions using the question panel on your GoToMeeting website. Or you can raise your hand and I can open your mic at the end of the webinar. Some of the questions I will be able to get to during the webinar, but others I will return to you after the webinar via email. You can follow us on these websites. You can also view a recording of the webinar on YouTube. So the key topics for today are how to read in sequence data into ICM and extract sequences from the PDB, how to create new sequences and edit them, how to map Uniprot and other annotations onto a sequence, and then once you've read in the sequences into ICM, how to then generate an alignment of those sequences. And then we'll show you how to edit these the alignments. And then one of the powerful things about the linking is that the alignment and the sequences are fully interactive with the 3D environment. So you can quite easily find conserved regions on your protein structures and link those directly to the alignments. These are fully linked. So you select here and it will propagate to the alignment, selecting the alignment, and that will propagate to the structure. And then we'll show how to display sequence conservation in the ligand binding pocket and calculate sequence identity and similarity in an alignment. And we'll end with just how to perform a blast search in ICM. So ICM has a direct link to a variety of the common databases. One probably most people are familiar with is Uniprot. So you can type in a Uniprot code or accession number and it will download the sequence. We also have links to ProSite, which is a protein domain and family functional site, and also PFAM, which is a database of curated protein families, and you can read in the alignments from there. So we're going to start with how to read in sequence data. We're going to start with looking at the example of the SH2 domain, which is a structurally conserved protein domain contained within the SRC oncoprotein and in other signaling proteins. The SH2 domain allows proteins containing these domains to dock to phosphorylated tyrosine residues on other proteins. And so we're going to use these sequences and read in some examples. So we're going to read from Uniprot. We're going to read in an alignment like this from PFAM. We're going to extract sequence data from the protein data bank. And we'll also create new sequences. So when you open ICM, you will see something like this. We provide a free 30-day license key. If you don't have ICM, just contact me and I can help you set that up. You may see different menus depending on what product you have, but the key ones we have today are the bioinfo menu. Okay, so how do we read information from Uniprot? So we first go to the search tab shown here, and we use the drop-down button. So in this drop-down button, we have all the databases that ICM links directly to. So we have the chemistry like PubChem, Drug Bank, and also the Protein Data Bank. But the ones we're interested in today are Uniprot. So you type Uniprot, and then you can type in a Uniprot ID or code related to a variety of different fields. So I'm going to use protein name. I'm going to look at look for a SH2 domain in the ABL2 tyrosine protein kinase, and ABL plays key processes in cell growth and survival. And so we'll look at this ABL2 structure. So you can use the Uniprot ID, Uniprot accession code, gene name, and it will find sequences for you. But we're just going to enter in this example ABL2_human and press search. When you do that, you get a table, and in the table we have the Uniprot ID, the accession codes that are related to this protein, the full name, the sequence, and then also other annotation in Uniprot shown here. Okay. So you can see we've read it in. So in the ICM workspace, this is where we have everything stored. So far we just have one table, which is this table. We want to download that information, the sequence information, into ICM. We just double click in this row. You see now that we have another element to our ICM workspace called sequences, and it's downloaded the ABL2_human sequence. This is a hyperlink, will take you to the website, to the Uniprot website. So just double click here and that will load it. Okay, so now we can see that in the ICM workspace, if we expand here, we can see the whole ABL2 sequence. And usefully, when you read in that sequence, it also takes in all the sites from Uniprot. So all the information, all the annotation from Uniprot is in this section called sites. So if we just expand here, this plus button right at the bottom of the screen, then we see all the sites that are related to this protein. The one we're interested in today is the SH2 domain, so you can just double click on this site, for example, where it says SH2, and it selects the sequence, that region of the sequence here. So this is useful, but it's better to, if you're looking at sequences, it's better to use the sequence editor to visualize them. To do that, you read in the sequence as we have done here, and we go to Edit Sequence. And this opens this window. Hopefully you can see it relatively okay. This has all the, on the right hand side, has all the annotation from Uniprot automatically displayed. So it's all color coded, so motifs, this gold color, certain domains are in green, any modified residues, phosphotyrosine, are flagged. And so you can find them just by double clicking and it will take you to that region. So some of these have more detail underneath. If you expand, you can go to those regions. So we can toggle this display on and off in the sequence editor just by clicking on sites. That would undisplay everything or display everything. Or you can be more specific and just use the check buttons, and this just shows certain regions here. So if you want to save this sequence as an image for a paper or something, you can export as image. You click here, export as image, and you can save in JPEG, PNG, or vector image file. We can display the sequence in a single line just by clicking here. That's going to put all the sites. So you can see that this is just a single line representation. We can add additional sequences to this if we wanted to. We don't have any loaded into ICM at the moment, we just have the one, but you click here, and if you had additional ones, you could use a drop-down list and you'd have more than one sequence displayed. Transparent is just this coloring for the annotation here. You can toggle that on and off. We can zoom in to the sequence or zoom out. You can also vertical zoom out. You can toggle the sequence on and off, and also the ruler at the top, and show labels. It's up to you. Okay, you can also edit the sequence. So you can add your own sequence. You can delete. You have to click on Edit, Delete here, and then you can add your own sequence just using the keyboard. You can add a new site. So if you want to select a region, click here, New Site, give it a category. This is just just a random selection, so I was going to choose region and just give it a name and a bit more description if you wanted to. And then you can change the coloring of that region. Just go OK, and now you have a new site. It also should be listed here. So you can see this example. We now have it here in the you can check it on and off. So you can also edit pre-prepared ones. So if you right click, you can delete it, remove it. You can edit it, just clicking here, similar thing. Change the coloring or whatever you want to do. And also add more description or change the description. Go OK. So once you're happy with it, you can just do cancel and close, and that will save the changes. Or you just uncheck edit, and that will close it. Okay, so that's the sequence editor. Another way to read in sequence information is to extract it from the protein data bank. So if you have a PDB, so I have a PDB, go to the search tab, choose PDB search, and an SH2 domain structure it's got the code 4EIH, so I was going to search for that and just go no, and this displays the structure. And we can extract the SH2 domain, the sequence from this SH2 domain. I was going to delete this table because it's getting in the way. So we can extract the sequence from the protein structure by right-clicking on the sequence here and choosing Extract Sequence. And then you see we now have two sequences, and here's the sequence of our PDB structure. The sequence is annotated red and green depending on the secondary structure. So red is alpha helix, green is beta sheet, and this magenta is like a non-canonical helix. So you can select it, you can see it here. Another way to bring in sequence information to ICM is just to go to File, Open, and then you can download your own files in FASTA format and for alignments and sequences. Here, so you just choose the toggle here. So if you had your own file, you can read it in. So in this ABL2 sequence we read in first, initially, we showed that one of the sites is the SH2 domain. So if we just double click on the SH2, you remember it selects here. So we can extract this segment of this sequence to a new sequence by just right-clicking and choose copy selection as text. So this selects the whole SH2 domain. And then we can make a new sequence with just the SH2 domain by going to File and then New, Sequence, and I can just give it a name. And then we can just paste the sequence. Go OK. And now we see that we have a third sequence here in our list of sequences. So that's how you bring in sequence information. Now we're going to move on to how to align the sequences. So in this example, we're going to read in a few kinase sequences from Uniprot. We're going to read in some crystal structures of kinases from the protein data bank. We're going to extract all the sequences and build an alignment. And then we're going to look at the sequence conservation surrounding the ligand binding pocket. And then once we've done that, we'll have a new alignment of that sequence conservation. And we can also map that conservation onto a 3D structure, onto a surface, or onto a ribbon representation of your protein. So built into ICM, there are a number of different alignment methods. There's a default one which I shall show you which we'll use, but you can change and toggle between different ones if you want to play with different alignment methods. There's also a variety of different comparison matrices as well. We also inbuilt into ICM is a secondary structure prediction method based on sequence, which is a modification of the DSSP algorithm. So when you see these arrows and cylinders, it means that secondary structure has been predicted or assigned or extracted from the PDB. So it's either prediction or it's from the PDB. Okay, so the example we're going to do, I'm just going to go edit, delete all, and we're going to start with reading in some CAMK kinase sequences. So we go to Uniprot, search here, and we enter the code, PIM1. The first one I'm going to read is PIM1_human. Read it in, just double click, that loads a sequence into ICM. The next one is KCC2A_human. Similar thing, just load it in. Now we have two sequences. And the last one I'm going to read from the Uniprot is CHK2_human. And double click. So this double click loads the sequence in. If you want to avoid running using double click, you know the Uniprot ID, you can just actually go file load Uniprot, and that will read it automatically without having to read in this table. I was going to delete this table, these tables. Clarity. So now we have these three sequences, and just wanted to point out that everything can be automated as well. So everything you do here is dumped into the ICM workspace, so you can learn the ICM command line and automate sequence analysis in the ICM terminal. So we have these three sequences. So we're going to make a sequence alignment. So to align them, if you want to align all of your sequences, you can just right-click here. So you need to select the sequences that you want to align. You can use Control to select a non-contiguous list, or use Shift to select all of them, or just click using Control, and that will select all three sequences. So to make an alignment, you just right-click and choose on your selection, and you choose Align Sequences. Okay. And that will open the alignment editor on the right-hand side. So the alignment editor is fully interactive. You can select a sequence and shift it along using the cursor keys, just like this. We'll show you that later on. So this is a fully interactive environment. So now, for this example, we're going to read in some kinase protein structures, and they have the PDB codes. The first one is 2PHK. So just go to search and then PDB search 2PHK and download that structure. Okay, so we can see the kinase structure here. And if you look in the A chain, you can expand and you can see the sequence. So we need to extract this sequence so we can add it to an alignment. So to extract the sequence, we right-click on the chain and choose Extract Sequence, like we did earlier with the SH2 domain. And now we have an additional sequence here. You see it's called 2PHK_A, and so it represents the name of the protein and the chain A. And you can see that this sequence has been extracted because we now have this little image here, this icon showing this sequence is this one. The other kinase we're interested in is 1QL6. Okay, so PDB search 1QL6. And that's displayed here. So in a similar way, we will extract the sequence from 1QL6. So we right-click on the A chain and choose the option Extract Sequence. Go OK. All right. So how do we add these new sequences that we've read into ICM into the alignment? There's a couple of ways we could do it. We could make a whole new alignment, or you can just choose the new sequence, so the 2PHK, which is the one extracted from the first PDB we read in. We can just take it and drop it into our alignment, and that makes the alignment shown here. We can do the same with 1QL6. So we have this alignment. We now can see that we have 5 sequences in the alignment. So we now, if you expand the object, you can see we have another icon here, and this tells us that this chain in this PDB is linked to the alignment called ALN. This is ALN here, and the same should be here as well. So we have this structure is linked to the alignment ALN. That means if we make any selection here in the ICM workspace or in graphics, that selection is propagated to your alignment, so you can easily find that region of your protein. Right. So the aim here is to look at the sequence conservation around the ligand binding pocket in this kinase, in these 2 kinases, and the additional 3 sequences we read in from the PDB. So let's just going to put default display. That will add the alignment to the bottom. Okay. So in this example, we have these 2 chains. We have 2PHK, which is green, and we have 2PHK, which is blue, and 1QR6, which is this greenish color. So at the moment, they're not superimposed. So we need to superimpose the two pockets. We've shown this before in previous webinars, so you can go back there. But a convenient way to superimpose 2 protein structures is to double-click on one here in the ICM workspace, make a selection, and double-click on the other here. Go to the Display tab and click on Superimpose. That superimposes them. Okay. Right. So we need to look at the we need to look at the sequence conservation. So yeah, you can do this. We had a question about other chains. You can use any other chains you want. In this B chain, I think is just a small peptide. It's exactly the same principle. You just need to make sure you extract the sequence and everything. So in this example, we have ATP shown here, and I'm just going to undisplay some things just to make it clear. So if we display ATP, we can select the neighbors of this protein, the neighbors to the ATP. So we right-click on the ATP and choose Select Neighbors, and you can see green crosses. And so we can display those as stick if you wanted to. If you propagate to all atoms, just choose R. So that shows all the residues surrounding the binding pocket. And you also see, now we've made this selection, that the selection has also propagated to the alignment. So you can see, hopefully, that this certain region here is blue. It's highlighted in blue and shown here and here. So if we wanted to see how well this region is conserved with all our proteins, we need to edit the selection a little bit more. So in the ICM alignment, there's this hammer button. If you click on that, it brings up the Edit Tools panel here. And there's an option called Propagate to all sequences. So this is in the selection panel of this editing, and Propagate to all sequences. If you do that, we can see that now our selection from our protein structures, which were the bottom 2, has been propagated to all of the sequences in the alignment. So I was showing some annotation. You can box this region and color or whatever you want to do. But for this example, I just wanted to show that we can easily see the conservation in the pocket. At the moment, the pocket, we have this region here. We have a big gap, and another region here that's in the pocket, and a big gap, and another region here that's in the pocket. So we can hide all the regions that's not inside the pocket relatively easily. So in this editor alignment panel, there's a selection. So we can invert a selection. If we invert a selection, that will select everything outside of the pocket. like that, you see. So now you can see everything outside of the pocket is blue. And we have some options here called hide, and so what we're going to, and this will relate to a selection, a bit like in graphics. Anything you do with the display will react to a selection. So if we click hide, that's going to hide everything outside of the pocket. Okay, so now we have this, and this is an alignment just purely on the based on the sequence around the pocket. So you can see that we have quite a few fully conserved residues, as you'd expect in the ATP binding site. You can, just going to make this a little bit smaller. So it's fully interactive, so you can see where this lysine is, for example. If you double click in the alignment, it takes you to the lysine. And so it's a convenient way of immediately seeing where you may have some where it's not conserved, where maybe you could design, or design your inhibitor towards in order to gain specificity. So another way to do that is to, I was going to go back to the selection panel and choose unhide, and just display. So we can just display one of the chains, and we can color by the alignment. So at the moment the alignment is colored by, you can see in the editor panel, it's called by consensus strength. So fully conserved residues are darker, semi-conserved are in lighter yellow. So if we color the structure by conservation, we can go color by, and then alignment, and then the regions that are fully conserved. So you can see in this glycine-rich loop here we have a green patch, so that region is fully conserved in our alignment, and the whiter regions are less conserved. We can change that to different. We have it inbuilt into the editor. There's a variety of different options. You can choose, we're using consensus strength. Another option is, you find it, conservation. This gives a little bit darker coloring, color by alignment. So you can see that the coloring is propagated. Had a question about the selection. I made a selection initially around the ATP. There's inbuilt in this select neighbors. There's an inbuilt selection that does, I think, around 5 angstroms or something around the ligand. But if you want to be more specific, you can go to neighbors, and then you can add a radius. You can change the radius here. That will make a slightly different selection to the default. That was shown. So we can see the conservation by the coloring here. Okay. So sometimes representing the structure as a surface, you can see the pockets a little bit better. Next week we're going to be having a webinar on how to identify ligand binding pockets and allosteric pockets. But one way is to build a surface. So if we build the surface, if we make a selection and go to meshes and choose plain solid, you can see there's other options, but they take a little bit longer to run, but plain solid will do a reasonable job. And we can see the, if you go to view, sketch accent sometimes helps, gives a better perception of the depth. So we can see quite clearly the ATP binding pocket here, and other cavities around the structure. So you could use this tool to color the surface by conservation. So we can do that in different ways. One way you can see we go back to the selection panel here in the editor of the alignment. So we have some options. We can select by consensus. So uppercase X represents fully conserved. So if we click here, select, that selects all the fully 100% conserved residues in our alignment, and so it's made a selection here in the alignment, as well as in the ICM workspace. You can see these green crosses, which represents the selection. So we can color the surface by this selection. So when I made the surface, I made it for 2PHK, and you can see we have another object in the ICM workspace called G_skin. This is the surface. So you can toggle the surface on and off, basically. But to color it, we're going to make that selection again, and go right-click on the surface in the ICM workspace and choose color. And there's different colors, but we're going to choose, so you can color by sidechain type or other options. But we're going to color by atom selection. And I'm going to color the surface green, and the brush size is how big the painting is. Pretty something like 2 is reasonable. You can play around with different parameters there. Just go OK. So now we can see the surface has been colored by conservation. It's pretty reasonable. So if we wanted to select the semi-conserved residues, so for example, here we have an isoleucine in 4 out of our 5 sequences. That's pretty well conserved. Here we have charged residue, positively charged, 1 tyrosine, 2 positively charged. This one is defined as semi-conserved as well. So if we select these semi-conserved ones, we do a similar way. Rather than X, you can see that we have this hash, and hash means semi-conserved. So you select it. So now if we do the same thing, we can color, right-click, so color by atom selection, just choose yellow, and make the a bit smaller. Hopefully we'll see, hold on, select again, and then color by atom selection, and choose 3. Okay. Yeah. So now the surface is colored by. So if you have a sequence alignment, maybe your ligand, your drug binds to one species, for example, and not another, or is specific to one and not another, you could use this sort of thing to try and find why that happens. You may see some trends that you can use for structure-based drug design. Okay. So yeah, so you can also color. Somebody asked about coloring the surfaces. The surface, you have to right-click on the surface and then choose the options here. There's no option for selection. If you don't see selection, it means you haven't got a selection made. Okay. So before I move on to annotating alignments, another useful option is to be able to compare the sequence identity between each one of these structures. So how similar is CHK2 to PIM1, for example. So to do that, we can just right-click on the alignment, and then there's an option called Calculate Sequence Similarity from Alignment. So there's different ways of doing it, but if you wanted to compare all your sequences against PIM1, for example, you choose PIM1 as the reference sequence, or you can use drop-down and choose any of the other sequences in your alignment, and just go OK. So you can see now that we have PIM1 versus CHK2. You have the sequence identity here, and for each combination you can see. And then at each point in the alignment it's propagated to a table, and so you can see which residue numbering at each point in your alignment. So leucine 203 in this position, you can play around with it there. Sometimes also it's nice to be able to look at the alignment strength, how well conserved at each point in the sequence is the alignment. So to look at the alignment strength, you can go to Tools, Bioinfo, and then choose the option called Calculate Alignment Strength Profile. It gives the name. You can just go OK, and then you get a table with this. So this is the position in the alignment and how well this is conserved at each position. You can map this to the alignment as well by going into open up the terminal on the side, and you can choose plot, and then you get these bar charts shown here for the conservation. So the larger the bar, the more conserved at each point. You can change it from bars to lines in your alignment shown here. Okay. So this is the alignment, how to build an alignment. You can also annotate alignments and save them as images and other things. So we have an example. I had a few questions about the alignment method. You can change in Bioinfo when you do a multiple sequence alignment. Then you can change the comparison matrices here, and you can be more specific about some options for opening gaps and other options here. So you go to Bioinfo, and then you can show here. Okay, so I had some other questions I would try and get back maybe by email for some of those. So in the third example, we're going to start to annotate an alignment, so more user annotation. So in this example, we have G protein couple receptors, obviously a big drug target. They will share similar structure in terms of 7 transmembrane helices, but they lack significant sequence homology between subfamilies. In the future webinars, when we do modeling, we have a GPCR modeling example, and usefully for each helix, although they don't have a great deal of sequence similarity, each helix has some, usually in family A GPCRs, has some conservation motif for each one, so you can line up the helix correctly. So for example, helix 1, we have a glycine, and it could be any residue times 3, and then followed by asparagine. In helix 3 is a kind of famous motif in GPCRs called the dry motif, shown here. So this helps you build up the alignment. So in this example, I've read the GPCR alignment from ProSite. So the alignment is already made. Let's say and it's going to go here. So I just need to read the example. Okay. So here we in the graphics we have the GPCR structure, and the GPCR structure sequence has been extracted as we did before with the kinases, and so the you can see that it's here basically. It has PDB code 3P0G, and the structure is here. So this is a ready-made example. It's available on our website. You can download it. And it also has a number of other G protein coupled receptors in the alignment shown here. So because it's linked to the protein structure, we can expand here, and you can take some other structural information. So in the bottom of this editor panel, you can see an option called Show Secondary Structure. So if you click here, that will show the secondary structure from here propagated to here. So red is helix, alpha helix, non-canonical helix, alpha helix here, beta sheet. There isn't any, it's just helices. So we can also show the profile, which is sometimes useful. If you click here, it opens this panel, and click profile, and then at the top of the sequence we have how, if this leucine is fully conserved, then you get L, the large L shown here, and then we can easily see the dry DRY motif in helix 3 here. So yes, so this is selected here. So for example, we can select a particular helix if we wanted to. So maybe, I think this is helix 1, we can select this region by clicking and dragging, and you can then, if you right click on any selection, then you've got some options such as draw box, red for example, or you can change the font, or you can shade a particular region, okay, and then set font color to a different color. It's not very good. We need to like this, or if you don't like what you've done, you can just select it and say clear, and that'll go back to the original selection. So there's options to change the font, if it's bold or not, you can change. You can also add your own annotation. So if you click and drag over a region, you can right click and you can say annotate region, and so you can give it a name, say transmembrane one, and go OK. So now you can see eventually, once it's done, yeah, you can see here that the annotation is propagated here. So if we select here, that selects that selection, and it will also propagate to the structure as well. So the selection here propagated here. Because we've added our own annotation, it's also listed in the structure as well, so you can find that region relatively easily. Okay. So Windows alignment main. Okay, so you can also you can see this has show secondary structure, and also says show disulfide bonds. So if there are disulfide bonds, they are shown as these yellow lines here, linking the cysteines. So you can click on them and select them, go back to the structure. You can see that the disulfide bond has been selected here, and you can display it in stick, for example, and find that region. Okay, so I had quite a few questions. I need to, some of them are not straightforward. I'll try and get to them afterwards, but we'll see. So we have so we selected the disulfides. We can also annotate in other ways. I showed earlier that you can add transmembrane, you can add your own text, but you can also annotate an alignment by residue numbers. Okay, so it gives the residue numbers for each one at this level. You can also annotate using shapes, sometimes useful for a figure. Okay, so you can see here has these options. Okay, so once you have a, you can also add a title to the alignment if you wanted to. Just click title here. Okay, so you can save this as an image by right-clicking and choose Save Image. You can save it, copy it to a clipboard, and paste it. You can also edit the alignment. I showed earlier, but if you select a region like here in the alignment, you can use the cursor keys to shift it. And you can see that the consensus coloring, everything is done on the fly. It changes that that region. Like so. So use the cursor left and right buttons. So sometimes it's useful to look at the tree as well, that's the underlying tree of this alignment. So you can do that by clicking tree here, and that shows the tree in this region. So you can change the distances of that tree. You can also use the tree to rearrange your alignment. So if you wanted your one structure to be on the bottom, for example, we can click it, you can drag it and drag that sequence to the bottom, and that will remake the tree. And so if you have a reference sequence that you always want here, you can play around with moving the sequences around here. Okay. So there's the tree can be toggled on and off using this button here. There's other options. So if you make a selection, so if I select here in the structure and I can't find it, so you've got very, very big alignment, you can flag that region. It's called highlight selection. So then hopefully in the GoToMeeting it shows you that that region is flashing red, so you can find your selection relatively easily. That's in the selection part of the alignment. You can choose view differences. That's sometimes an easier way of spotting the conservation in your alignment. You can view differences. There's horizontal scroll, which gives you the alignment in this way, and you can unalign the alignment. You can also change certain regions. So if you've got a very big gap, so if you have a gap of 50, it will hide that region just to clean up your alignment a little bit more. You have different options to flag, where you've hidden the gaps, shown here. Okay. So I just wanted to end with just to show you how you can do a blast search in ICM, which is helpful for when we move on to homology modeling and things like that. So simply to do a blast search, we can blast search this FFAR2 human GPCR. It's a target for anti-obesity drugs. So we can find templates for this sequence. So we go to To do a BLAST search, we go to Search, and choose BLAST, and somewhere here there's an option called BLAST, NCBI BLAST, and the sequence we're going to BLAST is FFAR2, so it should be in this drop-down list at the bottom here. And then we can choose what database we want to BLAST. Do we want to BLAST the whole of Uniprot, or do we want to BLAST search just the sequences from the PDB? So if you're looking for a template, one option is to do this, sequences from PDB, and just go search. And this will find the hits. So the search is finished. You should have seen a progress bar down here. Just go OK, and this will load in the BLAST search results. So now I have a table, and you can see the closest. This is quite useful for seeing how well your the coverage of your sequence to the template is, and these are ranked by similarity, and you can see the PDB code here, 5CKQ. I'm not sure if you double click it'll load that structure or not. No, it doesn't. So you can just read in this PDB code and it will display. Okay, so there's lots of questions. Some of them definitely have to get back by email, but I just want to thank you. I'll try and answer them in a minute after. So thank you very much for your attention and for your support of the webinars. We appreciate it. The next webinar is next week, and we'll be using the method ICM Pocket Finder to find ligand binding pockets and allosteric sites in protein molecules. Thank you very much.